Today you will learn about glycan analysis, why it's important, and how it's done. My name is Dr. Rodney Bannwart, and I am a principal scientist here at Emery Pharma. Prior to Emery, I did LC-MS analysis of biologics for 20 years at places like Alcon, Novartis, and Pfizer. A simple glycosylation on a monoclonal antibody is illustrated in the figure. mAbs are glycosylated at a specific arginine residue on the heavy chain. There are two heavy chains in the mAb, and both are glycosylated. The end release and labeled glycan determination is a detailed procedure that is useful throughout the whole product development cycle of biopharmaceuticals. The glycan profile of therapeutic proteins and mAbs can affect the protein's activity and function and its immunogenicity. The profile will change depending upon the health of the expression cell and is therefore monitored for lot-to-lot variability. The glycan profile is also important for biosimilarity. The profile is routinely elucidated in comparability and characterization studies as well. Indeed, the consistency of drug production is supported by the consistency of the glycan profile. Even though this is not the most sensitive technique, glycan information is often obtained from LC-MS mass analyses. In the case of the mAb, the information on the glycosylations G0F, G1F, and G2F is demonstrated. Subunit and peptide mapping analyses also revealed glycosylation information. Some of the major glycans are illustrated on this slide. They contain N-acetylglucosamine, mannose, fructose, galactose, and sialic acid. This slide only illustrates a few of the glycans that are possible. In this slide, I am presenting the protocol we use at Emery Pharma to release and label the glycans for the mAbs and proteins we study. In the workflow, the protein or mAb sample is first treated with the enzyme PNGSF at 37 degrees for 2 hours. The enzyme cleaves the glycans from the asparagine residue in the protein and creates a mixture of both the protein and the released oligosaccharides. The next step isolates the saccharides from the protein mixture using solid phase extraction. I prefer to use a Hillock phase in this step. After elution, the glycans are dried by lyophilization and are derivatized with compounds such as 2-AB or procainamide prior to LC-MS analysis. On this slide, we have illustrated the labeling reaction of the saccharide. Chromatographic parameters that I prefer are presented here. The 2-AB glycan libraries are commercially available and are injected into the same UPLC MS system as the samples. Because the vendor has identified the main peaks in the library, the glycan assignments in the sample are simplified via retention time comparison. Fluorescence detection is very sensitive and is recommended for glycan analysis. In this slide, I have presented the LC-MS chromatograms of the 2-AB labeled glycan library on top and the results from our case study on the mAb on the bottom. In the mAb, we identified over 25 glycans. In conclusion, the end release and glycan labeling LC-MS analysis is the most sensitive and accurate method for characterizing the glycan profile of proteins and mAbs. Since the glycan profile can affect the activity and function of the mAb, its importance cannot be neglected. This analysis is useful from early development up through to commercialization and is included in comparability and characterization studies. While our work utilizes a modern high-field Orbitrap mass spectrometer, the method can be performed with a fluorescence detector, significantly decreasing the cost of the analysis but not the sensitivity. Thank you, and please contact us at emerypharma.com with any further questions and/or inquiries.